Okay, so now that we've established that DNA is indeed the genetic material, we're going to talk about the structure of DNA and how we figure out its sequence. We're not going to go into hyper detail about this. There are new, more advanced sequencing techniques, but I will briefly describe how we figure that out. Also, likewise, we're not going to go into vast detail about all of the work that went into discovering the structure of DNA. We'll just talk mainly about the structure itself. Crick and Watson proposed the model for it, but they didn't actually do any experiments per se. They were chemists that used chemistry rules to work this out. And there were a lot of other people that contributed to this, although they might not get the credit they deserve. So we'll talk about the structure of DNA, talk a little bit about ways of examining DNA, particularly Sanger method for sequencing it, and then talk about similarities and differences between eukaryotic and prokaryotic DNA, because there are some differences between us. Okay, so we've talked about this previously, but it's good review here. So DNA, one of our macromolecules, is made up of nucleotides. A nucleotide is a base here. In this case, we're looking at a generic nucleotide. We don't know if it's DNA or RNA, but a nucleotide is made up of three parts. We have the phosphate, and we could have up to 3 phosphates. We've talked about ATP previously. ATP is indeed a nucleotide. We have the sugar part. This could be ribose in RNA or deoxyribose in DNA. Then we have the nitrogenous base. This is what's going to be the different letters that are in there, the information containing part of a nucleotide. So nucleotide structures, we're talking 1 to 3 phosphates. These are going to provide energy. We've seen ATP has 3 adenosine triphosphate that can power reactions. The sugar, deoxyribose in DNA, ribose in RNA. Here's the difference between them. This is missing an oxygen here, making it deoxyribose. And then our nitrogenous bases. We have adenine, thymine, guanine, and cytosine in DNA. There are pyrimidines that have 1 ring. Not going to really ask about this, but it is kind of important to the structure because a pyrimidine pairs up with a purine. So you'll see A and T pair together and G and C pair together in DNA. In RNA, we have a different base. Instead of thymine, we have uracil, which gets used, but it will pair with adenine the same way. Why? Why uracil and not thymine? That's how evolution has worked it out. And the system keeps working, so it has not led us all to going extinct. So that's my answer for why that is. I'm sure there are various hypotheses there, but I've not seen a convincing hypothesis that we definitively say this is why. So here's our structure, right? Phosphates, our sugar, and then the nucleobase here. Okay, I do need to point out that there are some important ways that we label the different pieces of this. So you'll see in this diagram here, I have the different carbons on the sugar labeled. I have the 1', 2', 3', 4', and 5' carbon labeled here. Organic chemists, they love to have ways to describe organic molecules, so they named it this way. Why do I point it out? Well, the 5' carbon and the 3' carbon are going to be really important to replicating DNA. We will find that new DNA nucleotides can only be added onto the growing chain on this 3' end. So DNA strands will grow in what we call a 5' to 3' manner. So that fits in perfectly with what I call my 3 rules of DNA. Okay, these are my 3 rules of DNA. If you ask somebody else what are the 3 rules of DNA, they'll be like, what are you talking about? Obviously, these rules are important, but I've kind of narrowed it down to 3 things that I think if you remember these 3 things, you cannot possibly screw up anywhere in any of the stuff we're going to talk about here. Okay, so rule number 1, complementary base pairing. A is going to pair with T, and G is going to pair with C. And I will show you a diagram of this in a moment. Don't worry. Rule number 2, DNA is built in a direction. it's built in a 5' to 3' direction. Okay, you can only add new bases onto the 3'. And finally, DNA strands are antiparallel. They run in opposite directions. So if one strand is going 5' to 3' to the right, the other strand will go 5' to 3' to the left, and they will pair together in the complementary base pairings. So let's look at a real model here. Okay, here's our DNA. This is the 3D model. We flattened it out. Okay, first part. DNA is kind of like a ladder that's been twisted, right? It's a double helix in here. So we have kind of the rails of the ladder on the side, or what I call the backbone, and then we have the rungs in the middle. Here you can see the complementary base pairings, A with T and G with C. Okay, how do these bases pair up? Well, it has to do with hydrogen bonding. Okay, so remember we talked about our different types of bonds. In this case, hydrogen bonding holds A and T and G and C together. And indeed, A and T will form 2 hydrogen bonds, and G and C will form 3. This is how the cell knows what to pair together, because we have a purine with a pyrimidine, and that fits properly. And we know it can make two hydrogen bonds, so it's got to pair with T, which can also make 2 hydrogen bonds. Whereas G and C, they can make 3, so they will pair together. Later, this base pairing will be important. I'll talk a little bit about the hydrogen bonds in just a sec, but know that those are pairing together. All right, how about direction? Rule number 2, DNA is directional. Okay, so here's a 5' and here's a 3' carbon. So this chain goes 5' to 3' down. Here's a 3' end. We could add a new DNA base here. On the other side, we have a 3' end. They're anti-parallel. So this one runs in the opposite direction, which is our third rule. So this one runs downwards. This one runs upwards. This will be important when we talk about DNA replication. All right, cool. We have those 3 rules. We have them set on the model here. So I said those hydrogen bonds were important. Hydrogen bonds hold the bases together. Now, hydrogen bonds are not super strong on their own. But when you get several million of them together in like a giant strand of DNA, they're actually quite strong. They're not as strong as covalent bonds still, but they are pretty strong. So this is important because this is going to allow DNA to unzip. And the more hydrogen bonds you have, the more energy you're going to need to put in to unzip the DNA. When we talk about PCR later in the course, a more advanced thing if you go on to learn about PCR at an advanced level, the more G's and C's you have, the more heat you got to put in to break apart the G's and C's. So A's and T's only have 2 hydrogen bonds. Less energy needs to go in to break those. Okay, so those are the hydrogen bonds that hold the complementary base pairs together. But what about the backbone of the DNA? This backbone of the DNA, which adds new nucleotides on, this is much stronger. It's a covalent bond, and we're going to utilize our triphosphates to power the reaction. So here is a base. It has a free 3' OH, where we can add a new nucleotide. Here's a new nucleotide, and it has these 3 phosphates here. Those are going to provide energy for the reaction. So to add this to the growing chain, we can only add it to this 3' OH. We're going to cut off 2 of these phosphates and stick these 2 molecules together. This is going to form a strong covalent bond. This is called a phosphodiester bond. It is very, very strong. It's much stronger than the hydrogen bonds that hold it together. So when the DNA unzips, the rest of the molecule stays intact. It does not fall apart. This DNA backbone is very strong. That allows things like ancient DNA to be sequenced because they're really, really strong molecules. All right, so how do we know that this structure is the structure? Well, you might say Watson and Crick discovered it. Yes and no. What they really did is they took a lot of other people's findings and they used them to build this model. They in no way confirmed this model was real through experiments. They just said, hey, the math seems to work. this is what we think the structure of DNA is. One of the pieces of data that they used was this X-ray diffraction image from Rosalind Franklin. So X-ray diffraction, it's a microscopy technique that shoots X-rays into things and they bounce off the atoms, and the way they scatter tells you about the arrangement of those atoms. If you're an X-ray diffractionist, this apparently tells you that it is a helical molecule, which was important. Rosalind Franklin died before Watson, Crick, and Maurice Wilkins got the Nobel Prize for this structure, and they don't give Nobel Prizes posthumously, so we'll never know if she would have been included in this. There's a lot of controversy around whether her work was used with her consent or not. She worked for Maurice Wilkins, or kind of with Maurice Wilkins. She didn't live very long. She worked with X-rays and ended up dying very, very young, so we don't have the full story, but there is a lot of friction in this part of the discovery of the double helix. Crick and Watson, on the other hand, they were chemists. They used data like Chargaff's rule and Franklin's X-ray images, as well as stuff Wilkins told them. And they took all of this together and they used chemistry rules and they built their model that you can see here. Here we have Crick and here we have Watson. Watson would go on to be a controversial figure who said some not flattering things about the evolution of humans and eugenics and things like that. But Crick is well loved in the scientific community, so only half the pair was controversial. But the model was borne out through many experiments. But it would take, I think, until the 70s to really nail down that this was indeed the accurate structure of DNA. And there were other people involved in this race. Again, if you're interested in this, I would talk all about this in an entire lecture in Micro 250. I haven't even touched on Linus Pauling, the Nobel Prize winning chemist who proposed a triple helix model and was racing them to figure out the structure. There's a lot that goes into this story. And what we're going to focus on, though, is that structure that they proposed, which I've shown you. And I've shown you the 3 rules that govern that. So we know the structure. What can we do with it? Well, for a long time, we were looking at DNA using a technique called gel electrophoresis. And this separates DNA based on size. Now, that might not seem super useful, but it can be because there are ways that we can differentiate the size of DNA from one person versus another, like DNA fingerprinting. So here we see a gel image. Here's the gel being loaded. And we apply an electrical current to this, and it causes the DNA to run through the gel, separating it based on size. Small pieces go farther than big pieces. And here we can see all these different fragment sizes. It looks like these two are maybe the same. Maybe they're from the same person or something, but all these other ones are different. So how does that help us? Well, let's examine what's going on here. In our gel, we have wells that are in there, and we load our DNA sample into the gel. The gel is made of agarose. It's a very fine, basically extracted from seaweed, a gelatin-like structure, and it's got all these little molecules, and as the DNA runs through, it bounces off those molecules. Really big, long DNA molecules bounce a lot, so they get slowed down, whereas short ones zip right through. It's kind of like a pachinko machine. If you know a pachinko machine's got little pegs and the balls fall from the top. Imagine a really big ball falling into a pachinko machine. It would bounce around a lot, whereas a really tiny ball would zip right through the pachinko machine. DNA has a negative charge, so if we apply a negative charge to the top and a positive charge to the bottom, the DNA will be pulled through. Okay, so then we can tell the size of DNA pieces. On the far left, we have what's called a marker lane. It's pieces that we know the size of. And over here, we have our different DNA pieces. How is this useful? Well, we could do something like PCR that tests to see if there is COVID DNA in a person. Okay, if there is, we make copies and we get something like a band of DNA on here. If there's no copies that get made, they don't have the virus. And like this lane here, there's no band. So that seems really simple. It's actually a little bit more tricky than that. We will do this in class, but we're separating DNA based on size here. And we can compare these different things, like these two pieces are the same. Maybe they came from the same person or something like that. So let's look at an example of how you would use that in, say, paternity. So we have two parents. okay? And they have a piece of DNA that has something in it that we call a restriction site. This restriction site in parent 1, the DNA will get cut if we apply a certain enzyme called EcoR1 to it. That will create a piece that is 7,000 bases long and a piece that is 3,000 bases long. The other parent does not have that restriction site. So if we put the enzyme in there, their DNA doesn't get cut. Okay, it stays this 10 kilobase long pair, 10,000 bases long. Okay, so their offspring should be a mix of parent 1 and parent 2, right? They should be a diploid. Each of them gave one copy, and we have it here. So how can we use this? Well, we could take all of their DNA and expose it to the EcoR1 enzyme. In parent 1, we run their results out on a gel. We should see a piece that is 7,000 bases long and a shorter piece that is 3,000 bases long. In parent 2, we should just see one piece, didn't get cut, that is 10,000 bases long. In the offspring, we should see a 50-50 mix of these two phenotypes, right? We should see half of their DNA not getting cut, so this band is half as strong as that one at 10 kb, and then half gets cut. So we see half is cut at 7 kb, and the other half is 3 kb. Parent 2, you are the father, right? This is establishing paternity. Obviously, you can't just do one site, you'd have to do many, but if you do a lot of these, you do 20, 30, right? That's a really good indication that if they all match up, that these people are related in some way. So gel electrophoresis, not only can we use it to say there's no band, there is a band, which we would use in like a PCR for a disease like COVID, we can also tell something about the sequence that's in there, right? This tells us that there is an EcoR1 site in there because it cut, and there's no EcoR1 site because it didn't cut. So that was our first attempt at figuring out what's inside the DNA sequence, okay? But really, it would be nice if we could just sequence the letters that are in there. Unfortunately, it took quite a while to figure that out. It wasn't until really the late 70s, early 80s, and really this became commonplace in the early 90s that we could read the DNA sequence. We call this sequencing technique Sanger sequencing. It is named for Fred Sanger, who basically worked out the process. And in effect, it's basically a very, very super dense gel that separates out DNA one letter at a time. And we have to use special DNA letters that have a fluorescent label on the end of them, and they will glow under a laser light. We'll talk more about this later. I'm going to show you an animation of this right now.
The first method of sequencing the genetic code was devised by Fred Sanger. To sequence the DNA, it must first be separated into two strands. The strand to be sequenced is copied using chemically altered bases. These altered bases cause the copying process to stop each time one particular letter is incorporated into the growing DNA chain. This process is carried out for all four bases, and then the fragments are put together like a jigsaw to reveal the sequence of the original piece of DNA.
Okay, so we piece together the sequence using Sanger sequencing methods, and we can actually use a laser to read all the different letters that are on there, and then a computer can put together the sequence that's there. We'll come back to this a little bit more in our biotechnology chapter at the end of the course, talk about some more advanced modern sequencing techniques. The Sanger sequencing still works, but it's a little bit limited. We get about, let's say, 900 bases from a good sequencing run. That sounds great, right? I could read 900 letters of DNA. Well, when you think that the average gene might be, over 1,000, you're not even getting a full gene there, and then the human genome is 3 billion bases long, then it starts to become a problem. And this is why the human genome took so long to sequence, because we were initially using these slow Sanger sequencing techniques. We now have much quicker sequencing techniques that could sequence a human genome theoretically in a couple of days, an afternoon, really, if you had enough machines together. This technique we'll talk more about in the future. So the DNA, it needs to fit inside of the nucleus, right? It's nuclein, as it was called by Miescher originally. And if you look at the human genome, there's about 2 meters of DNA that's there if you stretched it all out straight. How do you fit that into a microscopic cell? Well, in bacteria, the circular chromosomes get supercoiled. They swirl up and curl up this chromosome. But in eukaryotes, we need even more packaging. So DNA in bacteria is a circular chromosome. It gets coiled up by DNA gyrase. On the other hand, in eukaryotes, we have linear chromosomes that get wrapped around those histone proteins, and then the histone proteins get coiled up into what we call a nucleosome, and then we further condense the chromatin. And as we saw during DNA replication, the duplicated chromosomes get super, super condensed there. Eukaryotic DNA, when it is unwound, we call it euchromatin. Euchromatin is able to be expressed. The genes can be expressed on there. Whereas the tightly wound structure that's wound around those histone proteins, we call that heterochromatin. Okay, so before we finish, I want to again pitch my Micro 250 class. We run this every spring. If you find DNA and all this stuff really, really cool like I do, come take this class. Not only do we learn more about this in the classroom, but in the lab, we get to get hands-on, do biotech techniques, PCR, gel electrophoresis. We'll do that a little bit in this class, but way more in Micro 250. We do some genetic engineering. We make microbe art. We do all kinds of cool things. So if you want to get hands-on, come take that class. Okay, so we've traveled from Mendel through Sutton and Bovieri. Well, Miescher discovered DNA there. Sutton and Bovieri saying chromosomes are the genetic basis for Mendel stuff. Morgan and his fly room. Griffith figuring out the transformation experiment. But then we had Avery, McLeod, and McCarty explaining why transformation worked. Chargaff with his rules. Hershey and Chase figuring out, yes, DNA is indeed, Avery, McLeod, and McCarty were right. And Watson and Crick proposing their structure for the DNA double helix. We're not going to talk about this one, but again, Micro 250. And then the sequencing of the human genome in the 1990s. So it's taken a long time, and we're still working out what this all means. All right, let's finish with a review question. Okay, which end of the DNA strand can you add new DNA bases to? Go ahead and pause the video and think about that. So, you can add new bases to the 3' end. Here is the 3' end. You could add a new base there. Here is a 3' end. You could add a new base there. Okay. Hopefully you didn't pick Optimus Prime. Okay, just saying. All right. Nucleotides, the building blocks of DNA, are composed of 3 pieces: the phosphates, sugar, and the nitrogenous base. The sugar could be deoxyribose in DNA or ribose in RNA. The nitrogenous bases, there are information bit. We have A, T, G, and C in DNA, and uracil replaces T in RNA. The 3 rules of DNA. Complementary bases paired together, A with T, G with C. DNA is directional. It gets built in a 5' to 3' direction. You can only add onto this 3' end to grow. The DNA is antiparallel. So if you have one strand going one way, the other strand goes 5' to 3' in the opposite direction. Hydrogen bonds hold the complementary bases together, whereas the backbone of the DNA is held together by a covalent phosphodiester bond. Rosalind Franklin observed the helical pattern in X-ray data, and Crick and Watson used this data plus Chargaff's rule and several other observations to build the model of the DNA double helix that we now accept. Gel electrophoresis is a technique we can use to separate out DNA based on size. That was one of our first ways of investigating the sequence of DNA, was based on differences in size and where it would cut, where it wouldn't cut. If you want to read the DNA sequence, you got to use special DNA bases that stop the reaction. For Sanger sequencing, we saw those fluorescently labeled nucleotides that were in there. DNA is packaged by winding it up. In prokaryotes, we saw that it just wound around and around in the circle, just got wound up. In eukaryotes, we have it wound around histone proteins. Euchromatin is open, unwound DNA that can be expressed, whereas heterochromatin is wound up tightly and the genes are not being expressed. Usually that's found during cell division. All right, in the next section, we will get into how DNA is copied.